I'm Jennifer Isom-Schmicke, and today I'll be talking about the biopsychology of love and sex. I'll cover sexual development and abnormalities, sex differences in the brain between males and females, or the sexual dimorphism, human sexual behavior and sexual orientation, and then finally the neurobiology of romantic love. Humans have 23 pairs of chromosomes at conception. The 23rd chromosome pair, the sex chromosomes, determine one's genotypic sex. At conception, you'll normally have 2 sex chromosomes, making a pair, one donated by each parent. The mother, or the female, will always contribute an X chromosome, shown here in the picture on the right. The father can contribute either an X or a Y chromosome. The particular chromosome type that is donated by the father determines the genotypic sex of the individual. So, for example, if the father donates or contributes an X chromosome to the mother's X chromosome, then the genotype will be XX for female. If, however, the father donates a Y chromosome, the genotype will be XY for male, and that is the genotype that's depicted here in the picture. At conception, you will normally have 2 sex chromosomes, one donated by each parent. So these are the usual combinations, but they're not the only combinations. Very rarely, there are variations in these pairings that combine to form genetic abnormalities. The first one I'll talk about is Turner syndrome. Turner syndrome is noted by the symbol X and 0. And what that's indicating is that this individual, a female, has only a single X chromosome instead of the 2 X chromosome typical pair. So Turner syndrome is an example of a chromosomal abnormality. These abnormalities occur when an individual is missing a chromosome from one of the 23 pairs, or if the individual has an extra chromosome, so 3 copies for a pair. Down syndrome is one of those, that there are 3 copies for chromosome 21. Or when a chromosome structure is somehow altered by deletions or duplications. So for Turner syndrome, the fact that there is a X chromosome missing results in a female who appears to be fairly normal physically, other than being short in stature. The average height for women with Turner syndrome is 4'8". But this individual also has difficulties with fertility, and cognitive difficulties and cardiovascular disease is common. Another chromosomal abnormality is Klinefelter syndrome. Klinefelter syndrome is noted by XXY, which means it's a genotypically male individual, but that male has an extra X chromosome. Males with Klinefelter syndrome develop male genitalia, but the addition of that extra X chromosome causes some sexual characteristics for females to develop as well. So there may be breast development, as you can see in this picture. There is a waist to hip ratio that may be more consistent with a female than a male. Typically, the Klinefelter syndrome male has narrow shoulders. In addition to these physical abnormalities, the individual may also have cognitive difficulties, including lower verbal ability. Another chromosomal abnormality is the XYY genotype, and this has been referred to in the past as the supermale. So males with the XYY genotype have an extra Y chromosome. And what that results in is an individual who's taller than average, leaner than average, may experience lots of acne, may have minor physical difficulties as well. It was once thought that the addition of that extra Y chromosome contributed to an individual who was more likely to behave in violent and antisocial behavior. So it was once thought that prisons were overrepresented with this type of individual. However, the research has not really been supportive of this idea that supermales, or males with the XYY genotype, display any more antisocial behavior or aggression than males who have the typical XY genotype. Now I'm going to talk about sexual development and the 2 different types of development that can occur, organizing and activating effects. So the organizing effects in sexual development consist of those parts of sexual development that occur prenatally. And the interesting thing about organizing effects is that they are not reversible. They are lifelong in nature. The other kind of effects in sexual development are activating effects, and these are changes in sexual development that are brought on at puberty, and they are reversible. So with the right hormonal stimulus, these activating effects can be reversed. Both of these types of development are driven by hormones. And the first one I'll talk about is an organizing effect, and that is development of the gonads. So at conception until about 6 weeks of age, fetuses are undifferentiated. Male and female genotypic fetuses look exactly the same. Their internal sexual organs look exactly the same as well. So we would call them undifferentiated. Then at about 6 weeks, if you are genotypically male, the SRY gene from the Y chromosome will start the development of testes. The gonads will turn into testes, and those testes will start producing androgens, so testosterone, and other hormones that start to circulate in the fetus and defeminize the fetus. And further, they masculinize the external genitalia of the fetus. Now, the default for fetuses is female. So the XY genotype develops into a male with that stimulus of that testosterone. If there is no testosterone, the fetus will develop as a female. This slide shows the undifferentiated internal sex organs for males and females, and then the early development of sexual organs for male and female fetuses. For the first month to 6 weeks, every fetus looks the same in terms of internal organs. So this is depicted in picture A, undifferentiated. Male and female fetuses prior to 6 weeks of age have the equipotentiality to develop into males or females, depending on the hormonal stimulus. So in males, the SRY gene causes the testes to form from the gonads. The gonads then start producing small amounts of testosterone, and that testosterone, along with other hormones, starts to defeminize the fetus by degenerating the Müllerian ducts. Okay, here are the Müllerian ducts in the undifferentiated fetus. In the males, the Wolffian ducts, also shown in the undifferentiated fetus, start to develop into the internal male genitalia, the seminal vesicles, the vas deferens, etc. And testosterone masculinizes. In females, because there is no testosterone, the primitive gonads develop into ovaries, and those ovaries start to develop ova or eggs. The Müllerian ducts develop into a uterus, fallopian tubes, and the inner vagina. And the Wolffian ducts are absorbed. The external genitals are also formed. Just as we saw in terms of chromosomes, occasionally development goes awry at this stage, and abnormalities can result. One of these is androgen insensitivity syndrome, or AIS. This individual is a male, so the genotype of this individual is XY. However, androgen insensitivity syndrome is caused by a lack of androgen receptors. So even though the fetus was circulating testosterone, there were no receptors in that male fetus to be able to take advantage of that testosterone to masculinize the fetus. So what happens is you have an individual who looks like a female physically, but who actually has the genotype of a male. For these individuals, testes do develop, but since there are no receptors for that testosterone that's being secreted by the testes, they only partially develop male genitalia. Because a fetus without a hormonal stimulus of testosterone will develop by default into a female. Another potential issue in development is congenital adrenal hyperplasia, also called CAH. And CAH is a genetic defect, and it affects both males and females. It causes the adrenal glands to start secreting excesses of androgens. And those androgens are fine for males, but in females, they start to masculinize the fetus. And so the external genitalia sometimes are ambiguous. The female genitalia are enlarged. Now, when these children are born, even though they're females, their genitalia are ambiguous, and so often parents have to decide whether they want to raise the child as a male or a female. Surgery can correct some of this development, but if the masculinization is very pronounced in the female, the child can be raised as a male with additional reconstructive surgery. Currently, this is no longer a problem. Every child is screened for CAH at birth, and so medication can be given, and these children will live an ordinary life. Back to sexual development, organizing effects also occur in the brain. So there is a critical period for brain sexual development as well. So just to review, a critical period is a phase in an organism's life in which it's sensitive to a particular type of exogenous stimuli. In this case, it's hormones. And that stimulus or stimuli enable a particular kind of development. If the organism is not exposed to that particular stimulus during the critical period, that development will not occur. So in this case, there's a critical period for sexual brain development. And if the brain is exposed to testosterone during that critical period, then the brain will become masculinized. If it's not exposed to testosterone because it's a genetically female fetus, then that brain will not develop into a male brain. So for males, the testosterone that is secreted by the testes enters specific neurons in the brain of the fetus, where it is converted inside those neurons to estradiol, which defeminizes the male brain. Even though estradiol is the principal estrogen hormone, it is the testosterone that's converted to estradiol that actually defeminizes the male brain. In females, the development of the female brain also requires estradiol to develop normally. So this might lead you to wonder, why don't females with lots of estrogen circulating develop masculine brain structures? Why doesn't that estrogen get into the brain and masculinize it, like that testosterone that gets into the neural structures is converted to an estrogen and then masculinizes the brain? For females, there's actually a protein circulating in the bloodstream that binds with the estrogen that keeps it from entering the brain and masculinizing it. But moreover, what's more important is it's not free circulating estrogen in the fetus that gets into the brain and starts to masculinize the brain. It's actually testosterone that enters specific neurons in the brain where it's converted to estrogen or estradiol. So the brain becomes sexually dimorphic as a result of these different hormonal influences during this critical period. Now, dimorphic literally means two forms. Di is two, and morph means forms. So this indicates that the brains of females and the brains of males literally take on two different forms as a result of those hormonal influences. And these differences in the shape or the form of the male brain versus the female brain are what are referred to as sexual dimorphism. It's also called brain sex to some neuroscientists. Brain sex is a category that can be attributed to males or females, depending on what their brain looks like. So a male typically has a male brain sex, and females typically have female brain sex. These differences between males and females are so distinctive that a trained neuroscientist would be able to correctly identify a male versus a female brain. So how are they different? Well, some comedians have speculated. So this is not what I'm talking about when I'm talking about the sexual dimorphism of the brain, but it is kind of a fun way to look at the differences between males and females. There are actually differences in the architecture or the structure and in the function of male and female brains. So how are the brains of males and females different? Well, there have been lots of differences in structure or size that have been determined between males and females, and also some differences in neuron density and also in function. The ones that have received the most research attention are the corpus callosum, the frontal cortex, limbic structure, and the hippocampus. Those have been shown to be larger in females. The parietal cortex and the amygdala, which have been shown to be larger in males. And neuron density in the temporal and frontal lobes, which have been shown to be of greater density for females, which may explain why females tend to have an advantage in language processing. So one area that was particularly controversial was the corpus callosum. Researchers found that the corpus callosum in females was larger. So this led them to speculate that perhaps this is a reason why females are thought to be more intuitive. Maybe it's because they're able to access both hemispheres when they're thinking about things and use information from both of those hemispheres. Well, a follow-up study indeed confirmed that females do tend to have a larger corpus callosum. But when they looked at a slice of the corpus callosum and they actually counted the number of axons, and the axons, remember, are the wiring of the brain. It connects the neuron and sends the signals from the neuron to other neurons in the brain. So the axons are actually what are connecting all the neurons in both hemispheres. So when they looked at the number of neurons, they counted them in the slices of the corpus callosum. They found that, yes, although women's corpus callosum were bigger, they had the same number of axonal connections as men. So men simply had a more dense corpus callosum, but the same number of connections. So when we're looking at structural differences or size differences, it's really tempting to say that the size of a structure is probably related to the relative importance of that structure or how well that structure processes information. So for example, rats have a much larger amount of cortex devoted to olfaction or smell because that is an extremely important sense to their survival. Whereas primates have much more cortex devoted to visual perception because that's more important to our survival. So when they found that the corpus callosum was larger in women, it was assumed that somehow that gave women a structural and functional advantage, which in this case was not a very valid conclusion. So differences in structure have also been found for animals such as rats, particularly in an area called the SDN, which is the sexually dimorphic nucleus, and that's located in the medial hypothalamus, the medial preoptic area of the hypothalamus. Now, when rats are born, this structure shows no differences in size at birth between males and females. But size apparently is determined by exposure to testosterone during a critical period of sexual development. So it turns out to be 5 times larger in male rats. And they have also found that the size of the sexually dimorphic nucleus can predict the male's sexual activity as an adult. In the upper left corner of the slide is the SDN of an adult male. And this is an adult male that was exposed to testosterone normally during that critical period of sexual development. And you can see the size of the SDN POA, you can tell, is fairly large. The darker area indicates the nucleus. Right next to it, to the right, is the adult female. And this is an adult that underwent normal sexual development as well. So you can see there is a difference in the size of the SDN for the female, the adult female, and the adult male. So the adult male's SDN is 5 times larger than in females. Now if we go to this picture right below, this is a picture of the SDN for an adult female who was exposed to androgens during that critical period. So this female rat was exposed to testosterone during that critical period of development. And that testosterone masculinized the SDN, the sexual dimorphic nucleus, such that it looks very similar to the SDN of the adult male. Now right next to that is the SDN of a male rat who was exposed to DES during that critical period. And DES is a chemical that is similar in structure to estrogen. So the baby rat was exposed to this during that critical period. What it ended up doing was not allowing the masculinization of the brain to occur as it normally would. Now DES is kind of a scary substance because it was given to many, many pregnant women between 1940 and 1970 because it was thought to be a good thing to take when you were pregnant. It kept you from having birth defects and difficulties in your pregnancy. Then they realized that because it was so similar to estrogen, that it was having an impact not only on the health of the mother, but also the health of the baby. So it's been banned. But you can see the effect it had on the SDN of the rat. So what do all these mean? The findings overall related to the SDN indicate that the size of the sexually dimorphic nucleus is dependent on the amount of sex hormones that the rat is exposed to during that critical period of sexual development early in life. And they also find that the size of the SDN is highly related to that rat's sexual activity as an adult. So essentially, it's saying that sexual activity of an adult is related to those hormones that the brain is bathed in during that critical period. The interesting thing about that is that they find that for situations like in C, where you have the adult female exposed to testosterone, not only did she develop an SDN that looked very similar to the male's SDN, what they find is that rats in the situation also engage in sexual behavior that's very similar to the same sex. So, for example, this adult female, instead of engaging in female sexual behavior, such as assuming the lordosis posture that females assume to make copulation easier, or engaging in nesting behavior, this adult female would most likely be trying to mount other females. Now there is an analogous area in human brains to the SDN. There is no SDN for humans, but this area of the brain, also in the hypothalamus, the interstitial nuclei of the anterior hypothalamus, much easier said as the INAH, has been shown to be sexually dimorphic. In fact, it's 2 times larger in males than in females, so it's similar to the SDN. And what they found is that size may be correlated with sexual orientation, indicating that the larger the INAH is, the more likely the male is to be heterosexual in sexual orientation. So in addition to those structural differences of the brain between males and females, researchers have also found functional differences in the brains of males and females. So for example, lateralization of function is something that males and females differ on quite a bit. Male brains generally lateralize function earlier and more strictly than females. And by lateralization, I mean one hemisphere will tend to specialize in a certain function, like spatial processing or emotional processing, or left hemisphere specialization for language, for example, more than the other. And males will tend to specialize more than females will. So what does that mean? If you have a boy with left hemisphere damage, maybe a head injury, skateboard accident, that little boy may suffer from lifelong difficulties in language and reading, more than a little girl with the same head injury, because a little girl would have a better prognosis for recovery because females tend to use both hemispheres to process information more than boys do. So loss of function in one of the hemispheres, for girls or for females, it would be easier to use the other hemisphere to compensate for that loss of function. Consistent with this is the idea that more boys than girls are diagnosed with difficulties in reading and dyslexia. So that implies that if boys are more reliant on the left hemisphere for language and reading, and there is somehow dysfunction in that hemisphere, then they're going to be less capable than girls are of compensating with the right hemisphere to take on some of that function. As adults, it's been found that males have a much more difficult time recovering from strokes and regaining language ability than women do with a similar injury. And it's thought that's because women are less lateralized for language, so they're better able to use the right hemisphere to compensate. Other functional differences that have been found between women and men are emotional face recognition. Females have been shown to be better able to read faces and understand changes in emotional expression or tone of voice, and this is even as small children. So males and females have been shown to process things like emotional conflict differently. During emotional conflict, females tend to show increased activation in the amygdala, that structure in the limbic system that is known to be very involved in emotion, whereas males start secreting testosterone in response to viewing emotional conflict. Men and women also differ in their abilities in memory, emotion, vision, hearing, and response of the brain to stress hormones. Females have been shown to be stronger in language ability and understanding and memory for emotions. Males have been shown to have superior abilities in spatial processing. Now, this doesn't mean that there aren't women who are really good at spatial processing or men who are very empathetic or who have great emotional memory. It just means, on average, you'll find differences between males and females in these areas. Now, shifting from sexual dimorphism of the brain back to sexual development, just want to talk briefly about activating effects that happen in puberty. So sexual development at puberty occurs as a result of hormones that are secreted by the pituitary gland, and there are dramatic changes at puberty for both males and females. For females, there's the development of breasts and egg release during the menstrual cycle, and for males, the production of sperm, muscle development, hair growth, deepening of the voice, all of those things occur. For both sexes, there's a dramatic increase in sexual interest related to those sex hormones going into overdrive at puberty. So these changes at puberty, driven by hormones, are activating effects, which means they can occur at any time in life and they are reversible. As was the case during early phases of sexual development, organizing effects, there are situations in which development at this stage doesn't follow the usual pattern due to abnormalities. For example, the 5-alpha reductase deficiency. These are genotypically XY males born with ambiguous external genitals. This is caused by an inability to produce 5-alpha reductase, which converts testosterone into the form needed to develop external genitalia as a fetus, so in utero. So these fetuses aren't able to use the testosterone in the form needed to develop external genitalia as fetuses. Now, they do develop normal internal male sexual organs. As children, and at birth they appear to be female, and so they're usually raised as females. But, as I said, the internal male genitals are normally developed. And at puberty, when hormones set in motion the maturation of the external genitalia, surprisingly, many of these girls, in fact about 60%, mature into fully functioning males who develop normal external genitals, and they go on to live lives as normal males. So the next topic I'll cover is sexual behavior in humans.